Hello students, this is Tanika. Welcome to my channel PrepBiology. So today in this lecture we are going to discuss some numericals related questions regarding the Chargaff's rule. We have discussed in the previous class about the Chargaff's rules, what are the rules of the Chargaffs regarding the DNA structure. So today we are going to discuss some questions related to it. So the first question is if the double-stranded DNA has 20% of cytosine, calculate the percent of the adenine in the DNA. So in the double-stranded DNA, we have given the 20% cytosine is given 20%. So we have to calculate the adenine amount in the DNA. So according to the Chargaff's rule, we know that the number of cytosine is equal to the number of guanine. Okay, and also we know that according to the Chargaff's, the purines are equal to the pyrimidines. Okay, and if the cytosine is given 20%, then guanine is also 20%. Okay, so we know that they all can form the 100% bases. Okay, so we know that adenine we all have to calculate. Guanine we know that equal to the cytosine, that is 20%. Cytosine we are given, that is equal to the guanine 20%, and we have the thymine 100. So the total percentage of the guanine plus cytosine is 40. So around from 100%, when we subtract this 40, we have the 60% left base pairs. So these are the bases of adenine and thymine. Why? Because number of thymine is equal to the adenine. So we have the 30% about from the 60% adenine plus thymine. We have 30% adenine in the DNA and 30% thymine in the DNA. So what we have to calculate the percentage of adenine. So we have the adenine is equal to 30%. I hope this question is clear to you all. So this is the question from your NCERT exemplar. Okay, so this type of question have asked in your CBSE 12th board exams. Second question, read it carefully. While analyzing, this is the MCQ type question, while analyzing the DNA of an organism, a total number of nucleotide is 5386 nucleotides. Okay, so we have the total number of nucleotides is 5386 nucleotides, out of which we have the different percentage of the each basis. Adenine is 29%, guanine is 17%, cytosine is 32%, and thymine is 70%. So by looking these percentage of these bases, you have to considering the Chargaff's rules, it can be concluded that. So if we apply the Chargaff rule in this DNA having this percentage of the bases are present, so you all have to choose which option is related to the Chargaff rules. It can be concluded that. Okay, so we know that the Chargaff rule is for the double-stranded DNA. Okay, but here we have, according to the Chargaff rule, we know that A plus T divided by G plus C is equal to constant. Okay, so in this condition, we have the A plus T, that is A is 29%, T is 17%, okay, divided by G is 17% plus C is 32%. Okay, so this is not equal to constant. Okay, so this information is not applicable for the Chargaff rule, that is for the double-stranded DNA. So the answer of this question is it is a single-stranded DNA. Okay, so it is the single-stranded DNA answer. Why? Because it do not applicable, it do not follow the Chargaff rule, that is for the double stranded DNA. So that's why it is not a double stranded circular, or it is not a double stranded linear DNA. So the answer of this question is it is a single stranded DNA, because it do not follow this equation of the Chargaff's rule. I hope this is clear to you all. So next question is from your PYQs. Okay, this question have already asked in your previous year's class 12th board examination. So what is the question? The DNA fragment has 3000 nucleotides. Okay, so total nucleotides we have the 3000. Now out of which 160 are guanine. Guanine is 160 and total nucleotide is 3000. How many bases having double hydrogen bonds between them does this DNA fragment possess? So you all have to calculate the double hydrogen bonds. How many bases having the double hydrogen bond? So we know that the double hydrogen bond is between the adenine and thymine. So you all have to calculate how many these bases having the double bonds. Okay, so we know that according to the Chargaff's, adenine, purines and pyrimidines are equal in number. So we have this. Okay, and adenine are equal to thymine and guanine are equal to cytosine. Now if we apply this, that if we have the guanine is 160, so cytosine is also 160, that is equal to guanine. Okay, I hope this is clear to you all. So G plus C is equal to 160 plus 160 is equal to 320 bases. Okay, so this is the 320 bases of total G plus C. Now if we calculate the total nucleotides, we have the 3000 which is given. So for the bases of, to find out the bases of adenine and thymine, we subtract these nucleotides out of the total. So minus 320. So when we subtract, we have 2680 bases. So we know that the double bond, but we have to find that how many bases having double hydrogen bonds. So we know that the double bond hydrogen bonds are between the adenine and thymine. So we have to calculate the bases of adenine and thymine. So that is equal to the total number of bases in a given DNA fragment having double bond between 2680. This is the answer. Okay, so we have the answer D, which is the answer of this question. So this kind of questions have asked in your previous years. So make sure practice these questions repeatedly. So next question, this is the last question of this class, that understand it carefully. So next question is, a DNA molecule is 160 base pairs long. Okay, so DNA having 160 base pairs long and having the 30% guanine. So guanine we have the 30% and the total bases are 160 bases. Base pairs are present. Base is single and base pair, in one base pair we have two bases, adenine, thymine or guanine or cytosine. So we know that according to Chargaff's, pyrimidines are equal to purines. Okay, and we have the guanine is there. So guanine is equal to cytosine, that is equal to 30%. We know that guanine is equal to, number of guanine is equal to number of cytosine. We have the 30% given. So out of 100%, we have the 30% cytosine, 30% guanine. So 30 plus 30, 60%. When we minus, then we have the subtract, then we have the 40% adenine plus thymine. Okay, and 40% means adenine is equal to 20%, thymine is equal to 20%. Okay, so but we have to calculate the length of the DNA molecule. We know that the 160 base pairs. So this is the length of DNA molecule. So we have the 160 base pairs means, in 160 base pairs means how many bases? One base pair means two bases, adenine or thymine, cytosine or 1. Okay, so this is the 160 base pairs, means 160 into 2. Why? Because in one base pairs, how many bases are present? 2. So we have the 320 bases are present. So what we have to calculate, that how many adenine bases are present. So for the adenine bases, we know that the 20% of adenine is present. So we have the adenine bases are in the DNA are 320. This total basis, 20% of it, we calculate 320 into 20 by 100, 0 cancelled, 32 into 2 is equal to 64 adenine bases. Okay, so this is the 64 is the answer of this question. So this kind of questions have asked in your previous years. So make sure you practice these questions repeatedly. Okay, so this is the last question that I am giving you for your homework purpose, and the answer of this question, you have to calculate the expected percentage of base cytosine. Okay, so adenine percentage is given, so you all have to calculate the cytosine percentage. So answer of this question, please mention in the comment box. Okay, so I hope this lecture is beneficial to you all. This kind of numericals have asked in your class 12th CBSE board exams. Okay, so practice these kind of questions regarding the Chargaff's rule. If you like this lecture, like, share and subscribe my channel, that is PrepBiology. Thank you so much.